Hello, dear students. Welcome you back. I'm Dr. Arpita Dey. The topic for today's discussion is one of those topics from Ultrastructures of Cell Organelle. The topic for today's discussion is ultrastructure of mitochondria. Before we start, let's just recall our previous class lessons. When we were young, we were taught that mitochondria is the powerhouse of the cell. That means what? We know that this organelle is known to be the power generator of the cell. That is where energy is created or power is generated, by virtue of which we work. Okay, so we will talk about its structural characteristics. Fine. It is a double membrane bound structure. The outer membrane is about 60 to 70 angstrom in thickness and the inner one is about 50 to 70 angstrom in thickness. It has many plate-like infoldings into the cavity of mitochondria called mitochondrial crests or cristae. They penetrate the matrix of mitochondria. What is the use of having this plate-like infoldings? The main purpose is that these infoldings increase the surface area, which in turn paves way for more power generation. Now carefully observe the structure. These violet dots over here represent porine. These are proteins which are present on the outer membrane of mitochondria. It is followed by this matrix. It has the inner membrane which folds inside to form the cristae. The space between the outer membrane and the inner membrane is known as the intermembrane space. Okay, the cristae are studded with ATP synthase. These are the ones which form ATP, which we will look in the next diagram. These are calcium phosphate granules. These are ribosomes, and this is the DNA which mitochondria have. They have their own DNA, right? Now this is ATP synthase. One crystal has been magnified here to show the structure of ATP synthase. This contains the F1 particle, the F0 particle. Together it forms ATP synthase. Okay, this is the outer and inner membrane of mitochondria. Now we will see the space between outer and inner membrane is called Peri mitochondrial space, which is approximately 60 to 80 angstrom thick, and it is comparatively less in density. This matrix is also the membrane also extends in the crest part. This intermembrane space accordingly also extends in the crest. Within the matrix are small ribosomes, RNA, and one or more molecules of circular DNA, which is about 4.7 to 5.4 micrometer in length. It is filled with a relatively dense proteinaceous fluid material which is generally homogeneous in nature, but sometimes it may contain highly dense granules of about 40 to 50 angstrom in diameter. These are also known as intramitochondrial granules. Now we'll take a deeper look into the cristae. It can be divided into two types. Broadly, it can be classified into two types. One is septate, the other is tubular. The septate ones can be complete or incomplete. Okay, these are typical parallel-sided partitions appearing as triple-layered ones. The partitions or septa are composed of two unit membranes separated by a continuation of outer membrane. The next one is the tubular ones or the villus cristae. These appear as villi-like partitions of inner membrane being found in the mitochondria of protozoans, liver cells and neurons. These enfoldings of cristae provide an increased surface area within the mitochondria for enzymatic activity. The cristae possess F1 particles, as we have just now discussed, and coupling factors. Furthermore, the activities of several enzymes like succinate dehydrogenase, beta hydroxybutyrate dehydrogenase, rotenone sensitive, all NADH, cytochrome C, reductase, and ATPase, these are predominantly present in the cristae. I hope you all know the functions of these enzymes. It will be too long if I to discuss the functions, but I would urge you to study electron transport chain as a major function of mitochondria. You will come to know about each of these enzymes in that pathway. Most of these enzymes function in that electron transport chain which operates in mitochondria, those ATP synthase, everything will come over there. This particular lecture deals with the ultrastructure of mitochondria, so I'm not talking about its function and etc. in details. But to get it clarified, please do study the electron transport chain along with this. Now, these are believed to carry out oxidation reactions which supply electrons and also serve as a site for catalyzing of synthetic reactions with ATP. Each stop particle of inner membrane called elementary or F1 particle consists of a cuboid base. This is a little more structural detail of a stem and a long and round head. Please note the dimensions carefully when you are making notes from it. So this is all about the structural detail of mitochondria. It's a brief representation. You can always refer to books like The Cell, Albert Bruce. I always name this book as a good reference one. Other than that, you can look through, surf through the net. You can find out the structural details also. This lecture will give you a brief idea about the structural details of mitochondria, and I'm again mentioning, please do study the functions also in order to understand the structure in a better way. Okay, and feel free to ask any questions in the comment section. Thank you so much for your kind attention.